Well, nice to see so many people here today. My name's Michelle Burford. I'm from Griffith University. I was involved in helping organise the last meeting, and these are certainly really interesting meetings, and it's great to have the mixture of research and managers all talking about the cyanobacterial issues, and I think the last talk was a good example of some of the challenges we face. This session is called Environmental Impacts and Controls of Cyanobacterial Growth and Toxin Production. And our first speaker today is from Tokyo, and Manabu Fujii, and he'll be talking about toxin production in microcystis. Thank you very much.
Thank you. Okay, nice to meet you. I think I'm only Japanese in this room. Are there any Japanese in this room? Okay. Why I'm here? Because I was a postdoc in the UNSW maybe 10 years ago after I get my PhD. And I have a collaboration with Professor David Wade, sitting there. And I still have a collaboration with him, and he forwarded the information to me, and it's a good chance to me to have this kind of talk in front of you. So today I'd like to talk about the toxin production and why toxin is produced. It's a very challenging topic, and I do not have a specific conclusion, but I'd like to introduce one of my research topic. So in Japan, we also have the eutrophication and cyanobacterial bloom, particularly in the summer period. Nearby the Tokyo, we have several reservoirs, not only these reservoirs, but we have the several reservoirs, and it has the cyanobacteria or the blooms, particularly because of the nutrient input from the anthropogenic impact, particularly the agricultural land use. So, cyanobacterial bloom causes the deterioration of water, low dissolved oxygen, and in case of the drinking water reservoir, we have a bad odor taste. And the today topic, I focus on the toxin microcystin production. And in this reservoir, we measure the several sampling stations, and microcystin concentration is sometimes very high. It's more than 1 microgram per liter. But it's also varied depending on the stations and cellular content. That means the microcystin concentration divided by the cell concentration. It's also varied depending on the sampling station. So the question is why they have some kind of variations. Other study also has some variation. This is a long 1 year or 2 year series data. It has wide difference, wide variation of the microcystin toxic proportions. So the question is, it still remains unclear why and when toxins are produced, sometimes toxic strain, sometimes non-toxic strain. The why we have this kind of toxic and non-toxic strains. And also understanding of timing of toxin production and its mechanism is quite useful to maintain healthy ecosystems and also reduce the human health risk for sustainable water use. We know that the structure of microcystin, we have more than 200 or 250 variants, but we know basically we have ADDA structures and cyclic polypeptide structure. This is a general form of microcystin. You have different amino acid in the structures, but generally we have this kind of general structures. We also know the molecular level of the microcystin synthesis, MCYA, BC, blah, blah, blah. These genes are responsible for the microcystin productions. But the question is, again, why microcystin, this kind of molecule, is produced, and can we control this kind of microcystin production in the freshwater system? This can be the milestone paper, which show the ancient origin of microcystin producing genes. So you can see here phylogenetic the trees. You have a high similarity between the housekeeping gene and the MCY gene. So this indicates that microcystin gene is already exist when the cyanobacteria appear on the Earth. So probably a few billion years ago. So appearance of animals and zooplankton on the Earth is definitely far later than cyanobacteria appearance. So that indicates that so microcystin has microcystis, or cyanobacteria had a microcystin before the eukaryotic organism appeared. So these evidence suggests that just a coincidence of microcystin having a toxicity against animals or a defense to the predator is also less likely. So then, what is the role of microcystin? There are many functions or roles are suggested. Not only these three, but I pick up three major roles. One is extracellular, intercellular transport, cellular membrane channel. So microcystin can be incorporated in the membrane channel. Protein is here. A microcystin can be integrated in these protein structures. Or maybe microcystin is similar structure with siderophore, so it can bind to the nutrient metal like ions to help the intake of these metals. The other study indicated that protein binding molecules, radical scavengers, to prevent photosynthesis protein from oxidative stress. So, for example, you have a microcystin product structure here, and this kind of structure has an interaction of a thiol group in the protein, which can protect the oxidation of the protein. Or maybe the ADDA structure can react with reactive oxygen species and to scavenge or to regulate their concentration. The other role may be the signal molecule in column sensing, colony formation, or cellular recognition. So this microcystin can be the material that can regulate the cellular density or morphology. So these kind of functions are suggested. And just I'd like to introduce one of our study to examine the first hypothesis, which is related to the ion uptake. So previous researcher, about 20 years ago, they suggested that maybe microcystin is an ion transporter or ion storage peptide, because this conclusion is basically based on the toxic strain has a higher ion accumulation or higher ion uptake rate. So they suggested that microcystin is important in the ion intercellular transport. But this study has two different strains, so strain-specific variation was not considered. So we revised the approach by using the toxic mutant. So we use PCC7A06 with lack of MCYH, which do not produce the microcystin. And we try to examine the ion uptake kinetics in the toxic strain and the non-toxic mutant. And basically the result is the same. If you calculate the uptake parameters, half saturation constant is here, and maximum ion uptake is here. And the toxic one and the mutant it's quite same. And in case of PCC 7005 non-toxic strain even has a higher maximum uptake. So we also find strain-specific different. Actually, non-toxic strain has a higher iron uptake. So from this study, we think that iron uptake by microsystem maybe not less likely. And other study also show that multiple function of siderophore, including redox shuttle, reactive oxygen scavenger, and signaling molecule, which is published in the PROS 1 2012. And they concluded that it appears that catecholates siderophore can behave as a protectors of oxidative stress. These results support the idea that siderophore can have physiological roles aside from those in the iron acquisition. So this say that multifunctional roles of the siderophores. So yeah, microcystin may have also multifunctional roles, but maybe iron uptake function is less likely in case of microcystin. So we focus on the second the functions in the next study. So I'd like to just share about what is oxidative stress. And oxidative stress is defined as a disturbance in the balance between the production of ROS and antioxidant defense. And this reactive oxygen species can be produced in the cell due to the environmental stress like high light radiations or nutrient limitation like iron. And increasing reactive oxygen species can lead to severe cellular damage and death. Recently, one of the papers suggests the importance of microcystin in the protection of the redox reactive proteins like thioredoxin and peroxyredoxin. And actually, microcystin can bind to the peroxyredoxin under high oxidative stress conditions, and it can protect this kind of important protein. So, yeah, cyanobacteria may produce microcystin to mitigate reactive oxygen species-associated damage via binding protein or even scavenging the ROS. And we conducted a field survey to collect algal samples nearby the Tokyos, where we have eutrophication and algal bloom. We collected samples and tried to measure the cyanobacteria taxonomy and microcystin and metal species and DOC and pH. And then laboratory experiment, we focus on the oxidative stress, which I will explain. In the reservoir, we detected the microcystis is a dominant species based on the 16S RNA sequence. And again, yeah, we have a very different concentrations of microcystin inside the cell as well. So why did the cellular microcystic content vary? Any relation with environmental parameters? So we try to do the statistical analysis. In this case, principal components analysis. The MC is microcystin cellular content, which is positively related to zinc concentration and somehow copper, and negatively with magnesium, sodium, and calcium. And simple correlation analysis indicate that microcystin has statistically significant with zinc, sodium, and magnesium. So these trace metals may be associated with the microcystin production and perhaps oxidative stress. So yeah, zinc is a heavy metal, and if you have a high concentration of zinc, it can inhibit the calcium uptake. It has been reported. But magnesium, sodium, it can be competing. for zinc uptake, so it can reduce the zinc toxicity to cyanobacteria. So this kind of metal availability related to microcystin production and oxidative stress. So we conducted a laboratory study, incubation assay, using the microcystis PCC7806, and we use flag whale culture and calcium, magnesium, copper, and zinc, which can be related to the microcystin production, is valid in 4 levels. The second one is close to the original culture, and we increase or we decrease by several fold. And we measure the microcystin by ELISA and oxidative stress by using DCFH, and we also measure the cellular number. And to measure the DCFH, so oxidative stress, we use DCFH, which can penetrate to the membrane. And it can be hydrolyzed by the cellular esterase, and once hydrolyzed, it can form the fluorescence molecule after the reaction of reactive oxygen species. So the fluorescence intensity can be proportionally related to oxidative stress. And we found it, the kappa concentration increase, if it is increased, the oxidative microcystin concentration, this dot also increase, and zinc as well. And in case of nutrient metals, the less nutrient metal, you have a higher microcystin concentration and also higher oxidative stress. And if we plot this data, so the x-axis is the fluorescence intensity is related to oxidative stress, and y-axis is microcystin concentration. It has a linear correlation. So this shows that oxidative stress has is related to microcystin production. So the time is already 16 minutes. So this is some discussion about the heavy metal and oxidative stress. Excess concentration of heavy metal can inhibit the enzymatic activities and production of, and cause higher production of reactive oxygen species. I think I need some references. But in case of nutrient metal concentration, if their concentration is low, it also can cause some oxidative stress. But more mechanistic understanding in molecular level is needed by using the transcriptional and proteomic study. Okay, so this is the conclusion. So time is already 70 minutes, so I want to close my talk. Thank you very much.
So we have time for a couple of questions. Nick?
The recesses in my mind, I remember an old study that was done on...
Can you just use the microphone?
Yep.
Thank you.
So I remember some Israeli guys did a study on production of exopolysaccharides as a response, as an adaptive response to photoinhibition. I wonder if you've looked at to see if there's any correlation between exopolysaccharide production and toxin production.
Thank you very much for your question. Yes, I think polysaccharide has several functions as well for cellular physiology. For example, it can accumulate the metals on the cell, around the cell. Some study indicate that polysaccharide production, if you have a higher polysaccharide production, you have metal accumulation around. So it may be good for cell metal uptake in case of nutrient metals. So in case of from metal aspect, polysaccharide has these kind of functions. But if it also accumulate toxic heavy metals, so I'm still not sure it's positively and accumulation of metal nearby the cell are positively and negatively affect to the cell. Yeah. So this is one of my answers.
Another first question? I have one. You talk about zinc toxicity, but you didn't talk about the concentrations of zinc and if they are likely to be at levels where they would be toxic.
Yes. Thank you for questions. So, for example, zinc toxicity, well, in the natural environment, we have from 0.01 to 0.2 micromolar, and we use for one order magnitude higher concentrations. Of course, it can be related to the ligand concentration. It can affect to the free metal concentration. But what I can say is the zinc concentration or copper concentration that we employ in this study is, at least 10 times higher than environment.
Thank you.
Yeah.
All right, join me in thanking our speakers.